Hello and welcome to another video lecture on my YouTube channel. In this lecture we are going to discuss about the mechanism of acid secretion in the stomach. Hydrochloric acid is primarily secreted by the parietal cells present in the gastric glands of the stomach. And this hydrochloric acid is very important for the digestion. It prevents the colonization of the bacteria in small intestines, and it also activates the pepsinogen, and very important endopeptide so far as the digestion of proteins is concerned. And in this lecture we are going to discuss how these parietal cells are secreting these hydrochloric acid at the molecular level, and what are various paracrine factors or the hormones which are involved in the activation of these parietal cells to secrete this hydrochloric acid, and what are various chemicals which have the capacity to block this secretion from the parietal cells. And first let us try to understand the localization and distribution of these parietal cells within the various segments of the stomach. As you know, we are having four segments in the stomach, that is cardia, fundus, main body or corpus of the stomach, then we are having the pyloric part. Just at the opening of the stomach we are having this cardia present in the gastroesophageal junction. It is near gastric esophageal junction. So here we are having this cardia. The next segment towards the left of this cardia we are having somewhat dome shaped structure above, and towards the left side of this cardia region, that is superior to this cardia region, we are having this segment of the stomach that is called as the fundus. And inferior to this fundus we are having major portion of the stomach called as the body or the corpus. Segment is the pyloric segment, and this pyloric segment is divisible into three parts. One part is this triangular pyloric antrum, and this pyloric antrum here leads into the pyloric canal, and pyloric canal then leads into the pylorus, and pylorus then enters into the duodenum guarded by the pyloric sphincter. So these are the four segments of the stomach. These four segments have variation in anatomical features, and therefore we observe heterogeneous distribution of cells in the gastric glands of these four segments. That is, we are having different kind of cells in the gastric glands of these four segments. In the cardia region we are having the mucus secreting cells, and in this pyloric portion, pyloric segment, we are having the mucus secreting cells along with the chief cells which are involved in the secretion of the pepsinogen, and also we are having some endocrine cells like G cells or D cells which are secreting the gastrin or the somatostatin. And these cells are present in the gastric glands of this pyloric region. And in the gastric glands of these proximal parts of this stomach, like in the fundus and the body, we are having parietal cells also present in the gastric glands. That is, we are having parietal cells, we are having chief cells, we are having some endocrine cells, we are having mucus secreting cells. So far as the release or secretion of the hydrochloric acid is concerned, we are having parietal cells only in this portion, and so hydrochloric acid is secreted by the main body of this stomach and as well as the fundus segment of this stomach. Pyloric part of the stomach or cardia region of the stomach does not secrete hydrochloric acid because parietal cells are present only in the gastric glands present in the main body of the stomach or corpus segment of the stomach and fundus part of the stomach. Now here, as you can see, the distribution of various cells present in this gastric gland. At the base of this gastric gland, we are having these endocrine cells. These endocrine cells can be G cells or D cells, enterochromaffin-like cells, ECL cells. These endocrine cells will secrete their secretions like hormones, and immediately just below this gastric gland, we are having the capillaries, and the hormonal secretion from these endocrine cells will enter into this circulation. So these endocrine cells are present in the base of this gastric gland. Then we are having these chief cells. Chief cells will secrete the pepsinogen. And then we are having the parietal cells. As you know, parietal cells are involved in the secretion of the hydrochloric acid. And towards this neck region, we are having these stem cells or regenerative cells. Stem cells are continuously dividing cells. And then towards this neck region, we are having the mucus secreting cells present in this neck region. And on this surface, we are also having these epithelial cells which are involved in the mucus secretion. These epithelial cells will release their secretion in this channel, and this channel will lead the secretion towards the gastric pit. And this gastric pit is present at the surface of the stomach. These gastric glands will release their secretion in this channel, and this will finally enter into this gastric pit and then will enter into the lumen. And the secretion from this chief cells and parietal cells and this mucus cells is called as the gastric juice. That is, hormonal secretion is not the gastric juice. Gastric juice is usually the secretion from the chief cells, parietal cells and the mucus cells present in this neck region or this surface region. As you know, in humans, stomach releases almost 2 liters of this gastric juice in a single day. So far as the parietal cell activity is concerned, this plays a very dominant role to determine the composition of this gastric juice. If these parietal cells are not active, the composition of the gastric juice will be different from the composition of the gastric juice when these parietal cells are active. That is, these parietal cells are present in the gastric glands in two states. Either they can be at the resting state or they can be at the activated state or stimulated state. As we are supposed to explain the release of the mechanism of the acid secretion, let us try to understand what is the structure of these parietal cells and how this structure gets changed from the resting state of the cell towards this activated state of the cell. Here you can see the parietal cell. Parietal cell when it is in the resting state and parietal cell when it is the activated state. That is, this cell, this parietal cell here is in the resting state. That is the cell when it is not activated. This is the structure. This is the structural features of the parietal cell when it is not stimulated. Here you can see that this parietal cell morphologically is a triangular in shape. And here it is having these canaliculi. It appears that plasma membrane has been pushed deep into the cytoplasm to form these two channels. These are called as the canaliculi. This is the apical portion of these parietal cells, and this is the basolateral side of the parietal cell. If you can correlate here that this region of this parietal cell here, it is the basolateral, this portion, and it is the apical portion. This apical portion faces towards the lumen of this gastric gland. So we are having this apical side of this parietal cell and we are having this basolateral side of this parietal cell. In this apical side, we are having these two canaliculi, and these canaliculi are as if this plasma membrane has been extended deep or pushed deep into the cytoplasm of this cell, and this forms these two canals. And here the main feature here is that we are having these small vesicles present towards this apical side of this parietal cell. That is, there are various large number of these small vesicles present towards this apical side of the parietal cell. The membrane present in these vesicles is having proton pumps or potassium or chloride channels. That is, membrane within these vesicles is having proton pumps as well as potassium and chloride channels. And another feature of the cell, of the parietal cell, is this prominent nucleus placed in the center. Then we are having this large number of mitochondria present in this parietal cell, which indicates the functional activity of this parietal cell. So in the resting state, we are having large number of these small vesicles present towards the apical side of this parietal cell. Okay, when this cell is not activated, the gastric secretion will be having higher level of or higher concentration of sodium ions, and simultaneously it will be having low concentration of hydrogen ions or protons. That is, when the parietal cell is in the resting state, the gastric juice will be having the higher level of sodium ions and low level of protons or hydrogen ions. But when the cell is activated by some hormones or paracrine factors, what will happen? When this cell is activated, these small vesicles get fused with this plasma membrane of this apical region. During this activation inside the cell, we observe the increase in the calcium ions, and that calcium ions mediate the fusion of these vesicles into the plasma membrane of this apical region. And when these vesicles have chloride channels, potassium channels, and proton pumps in their plasma membrane, when these vesicles will bind, when these vesicles will fuse with the plasma membrane, they will cause two effects. First, they will increase the surface area of the plasma membrane in this apical region, and this increase in surface area will appear as the appearances of these microvilli. Second thing is that in this region of this plasma membrane, we will be having increased expression or increased appearances of chloride channels, potassium channels, and most importantly, the proton pumps. And these proton pumps will now be expressed in the increased proportion in this apical region of the parietal cell. So this is the parietal cell activated, or you can say the stimulated parietal cell. Now, when this parietal cell is activated and it increases its secretion, now it will increase the secretion of the hydrochloric acid as well as the intrinsic factor. Intrinsic factor, as you know, is a glycoprotein that helps us in the absorption of the vitamin B12 in the small intestine. When this cell is activated, we'll be having more proportion of this hydrogen ions and low concentration of Na positive ions, that is sodium ions. Here the condition will be opposite. When the parietal cell is in the resting state, then we are having sodium ion concentration in the gastric juice higher and low concentration of these proton ions. But when the cell is activated and it increases its secretion, then we are having hydrogen ion concentration in the gastric juice higher and low concentration of this sodium ions. Depending upon the activity of this parietal cell, we are having different pH in the lumen. When the parietal cell is in the resting state, the pH is between 4 and 6, and when it is highly active, then pH falls below 2. Now let us try to understand how the hydrochloric acid is secreted by this parietal cell. Very important point about the release or secretion of the HCl is that the hydrogen ion or proton is secreted separately and chloride ion is secreted separately. That is, there are two channels through which this hydrogen ion or proton is secreted separately and chloride ion is secreted separately. And the net result in the lumen is this HCl. Let us first try to understand how the proton or H positive ion is secreted by this parietal cell. As I explained here, that when the cell is activated, we have increased expression or increased concentration or increase in the number of this proton pumps, chloride channels and potassium channels. And here, as you can see, this is the proton pump, or you can say the hydrogen potassium ATPase. Here it is the potassium channel and here it is the chloride channel. And in this apical membrane, we have the increase in the number of these channels when the parietal cell is stimulated. Now let us first try to understand how the proton is released into the luminal side. The proton is pumped from inside of the cell towards the luminal side by this proton pump. On the luminal side, we are having pH equal to or less than 2. That is, pH in the luminal side is approximately 2 or less than 2. But the pH in the cytoplasm of this parietal cell is around 7.2. This is the difference in the concentration of the hydrogen ions between this luminal side and the cytoplasmic side is about 2 to 3 million times. That is, 2 to 3 million times the concentration of the hydrogen ions is higher in this luminal side than the cytoplasmic side. So to move this proton against this concentration gradient, this proton pump utilizes the energy in the form of ATP. So here you can say that we have to use ATP molecule, and this ATP molecule is being used to transfer this H positive. So one ATP molecule is used to transfer this H positive from the cytoplasmic side towards the luminal side. And simultaneously we are having the movement of potassium ion in the opposite direction. This proton pump or this hydrogen potassium ATPase, it transfers one proton or one hydrogen ion from the cytoplasmic side towards the luminal side, and at the same time it transfers the potassium ion from the luminal side towards the cytoplasmic side of this cell. Electrically it is neutral. That is, one positive charge is expelled outside the cell and another positive charge is moved inside the cell. So electrically it is neutral, but it uses one ATP molecule in this exchange of this potassium ion and this hydrogen ion. Why it uses one ATP molecule? Because the concentration of these protons in the luminal side is very high. So to move this H positive ion against the concentration gradient, this ATPase, this hydrogen potassium ATPase or proton pump utilizes one ATP molecule. So by utilizing one ATP molecule, it transfers this H positive ion towards the luminal side and simultaneously transfers this potassium ion from the luminal side towards the cytoplasm of the cell. And here this potassium ion transfers back into the luminal side through this potassium channel and it gets transferred passively. So through this ATPase or proton pump, it enters into the cell, and through this potassium channel it gets transferred back into the luminal side. Now what is the source of this H positive ion? That is, why from this H positive ion come? Here carbon dioxide and water gets enter into the cell. These two molecules combine with each other in the presence of an enzyme called as carbonic anhydrase. So in the presence of an enzyme carbonic anhydrase, the carbon dioxide and water gets combined to form this carbonic acid, and this carbonic acid then gets dissociated into H positive and bicarbonate. That is, carbon dioxide and water gets enter into the cell and these two combine in the presence of the carbonic anhydrase to form this carbonic acid. This carbonic acid gets dissociated into the proton and bicarbonate, and this proton gets transferred through this proton pump into the lumen of this gastric gland. Now you have understand the source of this proton and how this proton is getting transferred from the cytoplasmic side to the luminal side. And we have now H positive, or you can say the proton in the lumen of the gastric gland. Chloride is transferred separately. Here you can say this chloride, the cytoplasmic side is transferred through these chloride channels into the luminal side of this gastric gland. Now we are having the chloride in the lumen of this gastric gland, and we are having this H positive in the lumen of this gastric gland, and the net result of this combination is HCl. Now the question is, what will happen to this bicarbonate and what is the probable source for this chloride ion? So let us try to understand. Here we have, in this basolateral region, we are having this chloride bicarbonate exchanger. This is an antiporter. This chloride bicarbonate exchanger or antiporter transfers one bicarbonate ion from the cytoplasmic side towards the external side, and simultaneously what it does, it transfers this chloride ion from the outside towards the inside of the cell. Now this chloride ion is inside the cell, and this chloride ion can act as a source of chloride for this chloride channel. Now you can connect the performance of various channels with each other so far as the secretion of the hydrochloric acid is concerned. We are having this proton pump. This proton pump transfers one proton from the cytoplasmic side towards the luminal side, and simultaneously it transfers one potassium ion from the luminal side towards the cytoplasmic side, and this potassium from the cytoplasmic side gets transferred back into the luminal side through these potassium channels. And here the source is possible through the metabolic activities of the cell, and which involves the reaction between carbon dioxide and water through the mediation of this carbonic anhydrase, and it forms this bicarbonic acid. This bicarbonic acid upon dissociation releases this proton, and this proton acts as a source for this proton pump. And at the same time we are having this bicarbonate. This bicarbonate gets transferred from the cytoplasmic side towards the outside, and simultaneously chloride from the outside gets enter into the cytoplasm, and this chloride acts as a source for this chloride channel. And here it is the sodium potassium pump. This sodium potassium pump transfers 3 Na positive towards the outside and 2 K positives this inside. But so far as the production or the secretion of the HCl is concerned, we are having these pumps which play very important role. Another important point is that when this bicarbonate gets transferred from the cytoplasmic side towards the external side, it causes increase in the alkalinity of the blood, or you can say here we observe increase in the pH. pH of the blood is increased, so does alkalinity of the blood get increased. So this condition is called as the alkaline tide. This occurs in the blood. What happens in the lumen of the stomach? In the lumen of the stomach, we observe increase in the acidity. That is, we observe, so far as the pH is concerned, we are observing decrease in the pH value. So here in the blood, we are having increase in the pH value, and in the lumen of the stomach, we are observing decrease in the pH value. But this effect is reversed in the duodenum. For example, in the duodenum, we are having secretion from the pancreas as well as from the liver. And the secretion from the pancreas and liver, as you know, is alkaline. And it is this alkaline secretion from the pancreas and liver that neutralizes the acidic effect of the chyme. That is, when the chyme enters from the stomach into the duodenum portion, its effect is neutralized by the alkaline secretion from the pancreas and the liver. That you can say that in this portion we are having alkaline secretion, but in the stomach we are having acidic secretion. In the stomach we are having alkaline tide in this blood, but the blood in the pancreas as well as in the liver must be having simultaneous increase in the H positive concentration. Only then this alkaline secretion is possible. That is, when the pancreas or the liver is producing the alkaline secretions, and these alkaline secretions through ducts gets enter into the duodenum. Simultaneously we will observe increase in the H positive concentration in their blood. That is, so we can say that in the stomach we are having increase in the alkalinity of the blood, but in the duodenum portion we are having increase in the acidity of the blood, and in the lumen we are having this increase in the acidity of the lumen, but in this duodenum lumen we are having decrease in the acidity. So these two effects are opposite to each other, and they are well regulated, and they are effective in neutralizing this effect. So this alkaline tide is a temporary, it is neutralized by the increase in the H positive concentration in the pancreas, liver, blood. Now another point is how this release of HCl by the parietal cell is stimulated. Now let us try to understand how the stimulation occurs. Secretion of hydrochloric acid by the parietal cells can be stimulated by three factors. One is this gastrin, then we are having this histamine, and we are having this acetylcholine. Acetylcholine is released by the vagus nerve endings of the vagus nerve or cranial nerve 10th, and this acetylcholine is having the receptors in the basolateral portion of this parietal cell membrane, and these receptors once gets bind with the acetylcholine stimulates this cell through this signal transduction mechanism to release this hydrochloric acid. And similarly this gastrin released by the endocrine cells in the this gastric gland, this is having this receptor in the basolateral region, and then it stimulates this cell to release the hydrochloric acid. This is the direct pathway, that is this gastrin and acetylcholine when it directly binds the receptors present in this basolateral region. Another way to act on this parietal cell is through this enterochromaffin like cell. This gastrin as well as the acetylcholine is having receptor on this ACL cell, and once the gastrin is released or once this acetylcholine is released, it gets bind with this ACL cell receptors, and from this ACL cell we are having the increase in the release of this histamine. This histamine is having receptors on the parietal cell. Now the histamine released by these ACL cell is having H2 receptors, which are different from the receptors for the histamine involved in the allergic reaction. So here we are having different receptors for this histamine, and this through this receptor binding we are having the activation of this parietal cell that leads to the increase in the activity as well as the secretion of this parietal cell. So these three, histamine, gastrin, and acetylcholine are three factors which are related with increased activity of the parietal cells so far as the release of hydrochloric acid. Increased secretion of hydrochloric acid is observed when the gastrin, acetylcholine or histamine gets bind in the basolateral receptors present in this parietal cell, will cause stimulation, and this stimulation will increase the secretion of hydrochloric acid in the cell. Usually the release of hydrochloric acid is well regulated and well balanced, and sometimes under some specific conditions these parietal cells shows hyperactivity and accordingly leads to increase in the secretion of this hydrochloric acid, and which results in the increase in the acidity of the lumen of this stomach. There are various substances which are being used to block the activity of these parietal cells or to stop the activity or to minimize the hyperactivity of these parietal cells, and most commonly we are having two substances. One is called as the omeprazole and second is ranitidine. Omeprazole is a substituted benzimidazole, And this benzimidazole, or you can say the omeprazole, it covalently gets bind with the alpha subunit, that is cysteine residues present in the alpha subunit of this proton pump, and thereby it blocks the activity of this proton pump. And this ranitidine, it is antagonist that gets bind with this H2 receptor, that is histamine receptor, and by binding with this histamine receptor, it suppresses the activity or decreases the activation of this parietal cell by this acetylcholine or gastrin or histamine. So these two substances are used to block the activity or stop the activity, hyperactivity of this parietal cell in the abnormal conditions. So this is all about the mechanism involved in the secretion of this hydrochloric acid by the parietal cell, and it is now clear to you that these parietal cells present in this resting state, when it gets stimulated, it is secretes this hydrochloric acid into the lumen of this, and this secretion is stimulated by this acetylcholine, this histamine and gastrin.